Hello everyone, I'm Marco Gabrielli. I'm a postdoc at EAVAG, and for the last couple of years I've been working within the LeCo project looking at Legionella ecology. The LeCo project is a wide multidisciplinary project ranging from Legionella detection, epidemiology, management, and ecology. And today I'll present you my work titled Expansion, Restructuration, and Characterization of the Legionellaceae family. So, we all know that Legionella is the genus to which Legionella belongs to, for example, and is the solely recognized genus within the family Legionellaceae. And different legislation either target Legionella pneumophila or they can also target the whole genus, so Legionella spp. But while we know relatively well Legionella pneumophila, which is the most clinically relevant species, most of the other species are actually unknown. And so we asked ourselves, can we know something more about this species? Can we understand the ecological differences across the species and how they are distributed across the environment? And to do so, we took all the publicly available data that we could find. So we found about 5,000 genomes belonging to bacteria of the family Legionellaceae. And then we screened them through multiple steps to be sure that they were of high quality, and then we used good data. And this led us to obtain about 4,000 genomes, which unsurprisingly were mostly belonging to Legionella pneumophila because, of course, that's the most widely studied species. And so we use ANI, which is average nucleotide identity, to compare these different genomes and to cluster them within species, and by doing so we found 128 species and genomic variants or genome overs, and by doing so we obtained 403 genome overs. But once again, about half of them were belonging to Legionella pneumophila because again most of the genomes come from this species, and so we took only 4 of those, achieving a total of 164 genomes representing the diversity within the family at both species and genome over level. Then, once having these 164 genomes, we looked at different traits of these genomes. And first we looked at evolutionary history of all these different species. We then looked at the factors linked to host invasion, and then the metabolism. And finally, after all this, we linked them to sequences in 16S RNA amplicon surface to assess the environmental distribution. And all of this has been made in order to better understand the ecology of all these different species, which eventually could be translated to have and design better management strategies. So, starting with the results. Here on the right hand side, you see this tree which represents the evolutionary history of all the species that we found. And around this tree, you see this, let's say, crown, which represents the species information. So in red, we have the highly clinically relevant species. In orange, we have species that have been linked to clinical cases at least once. In green, we have known species, while in blue, we have the new species derived from the study. And as you can see, we have blue, so new species, both closely related to known ones, but also we have entirely new parts of this tree, so entirely new clades of new species that we found. And so this actually highlights how we know just little about this bacteria, and there's much more species out there that we know very little about. And then if we look at where those genomes were retrieved from, so the isolation source, we find that those genomes were found from both soil, a variety of aquatic ecosystems, and of course clinical samples. And it's interesting to note how for some species we only have clinical isolates, but we don't have any environmental isolates, while on the other hand there has been just one potentially reported cases of human to human Legionella transmissions. So we don't know where infections come from. But also at the same time, we see a lot of genomes coming from habitats which we wouldn't necessarily think when we think of Legionella, like for example, marine habitats. And so we need better information regarding the environmental sources of all these bacteria to understand where infections are coming from. And then we started to compare all those different genomes and species, and this time not using ANI, but using AI, which is average amino acid identity, which is a measure that is able to look at more distantly related species of bacteria. And by doing so, we find similarities down to only 44%, as you can see in this plot, which are values that are way below what is usually considered a threshold for genus level similarity, which indicates the fact that this family actually does not include just one genus, but contains multiple genera. And so, using these values, as well as the phylogeny that you can see again here on the right, we propose different genus level clusters, which interestingly also align with partly clinical association. For example, you can see that most of the orange and red species, so species which have been linked to cases, are in this red and blue genus cluster, while all the smaller ones on the right hand side, they actually contain no clinically relevant species. Which again, it's a first indication that all these different genus clusters actually contain species which behave differently, both clinically but also in the environment. And so to look more that into details, we start to look at, for example, the traits related to host invasion. So, and with host, I mean both the macrophages in my lungs, but also protists in the environment. And here we see once again the same phylogenetic tree, just this time in a linear form, not in a circular form. And we see again the species information and colors representing different genus clusters. And when we looked for the type 4 secretion system, type 4b secretion system, so the .ICM type, and the type 2 secretion system, which have been widely characterized for Legionella pneumophila for their role into invading, again, our lungs and protist hosts, we see that most species actually are characterized by having both of them. While these are not the only two types of secretion system, there's plenty of other secretion systems that are able to inject proteins into other cells. Like, for example, the type 1, the type 4a, the type 5, and the type 6. And as you can see here, all the different species actually have a different amounts of all these different secretion systems, which again might indicate that they're able to inject different proteins in different hosts. And for example, one thing that we found that was very interesting is that we found species which are actually characterized by a type 6 secretion system. And why am I highlighting that? Because the type 6 secretion system is usually believed to be used to, let's say, attack other bacterial cells to be able to then consume their content and, for example, to obtain nutrients in a nutrient-scarce environment. And interestingly enough, this is only present in a very few number of species within the Legionella family. And again, talking about scavenging and nutrients-rich or poor environments, now we look at metabolism. Again, same phylogenetic tree, but now we see three different metabolic characteristics which have been described for Legionella pneumophila. So the enteroendorenal pathway, which is linked to replication within the host, the synthesis of this energy storage compound called PHB, and the degradation of myo-inositol, which is a carbon source present commonly in different environments. And as we can see, all the species which are actually belonging to the same genus cluster, Legionella pneumophila, they mostly have all these characteristics as Legionella pneumophila per se. So they tentatively behave not too differently. But when we travel along this tree, we see now how species which are moderately different, they start to kind of lose all these traits. And species which are even further apart, they mostly have none of this, which indicates how these bacteria, they actually behave very differently than Legionella pneumophila because they have different metabolic capabilities. And for example, when we look at this genus cluster in green, we see how this has a reduced genome size. It lacks flagella, so the ability to swim in liquid environments, and again lacks this energy storage compound PHP, which are all signs that this bacteria might be purely intracellular and not facultative intracellular like Legionella pneumophila. And so all these different bacteria, they're not, let's say, interchangeable, but each of them has different traits, which leads to different lifestyles and different capabilities to eat and live in the environment. And talking about eating, we looked at the nutritional requirements of bacteria, because not all bacteria are able to produce and biosynthesize all the different essential amino acids, but in some cases they need to acquire them from the environment in order to be able to grow. And this is a characteristic which is called auxotrophy. So here in this heat map you see on the y-axis all the different genus clusters, and with one being the one into which Legionella pneumophila belongs to, while on the x-axis you see all these different amino acids. And let's say the more yellow a tile is, the less capable of producing that amino acid are the bacteria within that clade, let's say, that genus clusters. And you can really see how all these different genus cluster on the top part, they tend to be more yellow, so they are able to produce less amino acid required for their growth, while the ones in the bottom, they're more blue, so they're more able to produce these compounds. And interestingly enough, we see how all this, let's say, most of the bottom part of this plot is blue for cysteine, so it means that they should be able to produce cysteine. While actually when we are in the lab, we know that if you want to grow Legionella pneumophila, we need to actually add cysteine to the media in order to make it grow. And so when we looked at that with a bit more detail, we saw how actually Legionella pneumophila has the genes to go from homocysteine and serine to cysteine, which is what it would be called the pathway to produce cysteine, but it lacks all the genes to go from methionine to homocysteine, which is a required precursor. And so this is the reason why we need to add cysteine in our media. And also we see how there are some sulfur requirements which actually we are not controlling for, and those sulfur requirements are actually required to go from homocysteine to cysteine, which is again something that we might need to take into account. And so basically what this tells us is that if we want to bring these new species into the lab to study them better, we need to come up with new isolation strategies. We cannot keep using the same isolation media that we use for Legionella pneumophila, but we need to adapt it and to include for more and different auxotrophies. And so when we then look at how these species are distributed in the environment, we see how different species are actually differently distributed. Here, for example, to explain better this concept, we have on the top the indication of the genus cluster, so 1, again Legionella pneumophila, 5, 6, and so on. And then in the different colors, we see the proportion of detection of the given species within samples belonging to animal-associated environments, aquatic environments, plant-associated environments, and even within soil. And it's pretty clear that most species are actually able to live in all these different environments. The difference is that all these different species prefer one versus the other in a different way, because again, they have different metabolic capabilities and different traits related to infect different hosts. And finally, the other very interesting observation is that we see a lot of animal-associated samples, not just for Legionella pneumophila, but actually for a lot of different species. And so we wanted to look at this more in details, and to do so, we used the metadata, so the information of where the samples were taken from, which is actually what you see here. Here on the x-axis, you see all the different keywords that we found for that metadata, while on the y-axis, you can see how many times you actually have seen that word. And so we find first a lot of words related to just general human, let's say, associations like human, child, and so on, infection. But at the same time, we find a lot of other different keywords related to, again, human associated body parts, but which actually we wouldn't necessarily think of when we think of Legionella, such for example as skin, feces, and intestine. And in fact, in literature, there has been reports of a gastroenteritis caused by Legionella pneumophila. Finally, if digging even a bit more, we see how some other keywords are actually related to other animals, such as fish, mosquito, insects, and cattle. And those could actually be infected then by Legionella and could serve as a reservoir of this bacteria within the environment, especially in case of mosquitoes or fish, which are quite well distributed. So, to conclude, the family Legionellaceae, it's way larger than expected, and the species that are more distant to Legionella pneumophila are actually very different and have more limited metabolic abilities and tend to be more host associated. And finally, that most Legionellaceae bacteria are actually able to live in most environments, including several animal-associated ones. And before concluding, I just want to acknowledge all the people that have been included into this work and have helped to drive this work forward, such as Antonia, Alessia, and Frederick, and also all the researchers that have shared the data on public repositories, because without them, I wouldn't have been able to do this work. Finally, I want to thank you for your attentions, and be sure to check out all the other videos of the series to see what all the other exciting research that has been done throughout LeCo. And if you have any questions, please reach out to me. Here's my email address, and I'll be happy to reply to any other question that might come to mind.